भी चैट कहाँ पे देखनी है?
यूट्यूब पे।
ओके ओके ठीक है ठीक है। Good morning to everyone. Good morning. Starting in a minute. Just waiting for all to join in. So I hope I'm audible, visible, all of you. Good morning, good morning. Just a second. Okay, so we'll be starting the class in a second. I'm audible, visible? I'm audible, visible? Okay, so notes I've already shared. These notes I've already shared. So you can use it for your session. Yes. So this is the first session for the current online batch. Okay, and those who are watching from the last session also, welcome once again. So basically I'll be giving you a orientation, how to prepare, how to study, how to use the app and everything. Any doubt you can ask, and then I will be, yes, continuing with the class itself. Okay, so let's start with the basics. So basics is number one, you will pass the exam, 100% promise. Only thing is, just be sincere. Only one advice, just be sincere. Because I'll tell you, the truth is, most people who join online batches, they are not very sincere. The attendance, the number of tests, this is like, I can tell you, it's very poor. Okay, like hardly 20-30%. Clear? Because the fact is 20-30% are regular in class. 10% have given all the tests. All tests. And I'm not joking. I can show you all the data and you will say, yes, sir, you are right. Because the students are not, like giving all the things, they are not very sincere. So that's the biggest problem with the, online classes, that students, they, whenever they are not in the mood, they just quit the class and they will not be studying. So that's the biggest problem I find in the online batch. Otherwise, it's a very good medium. It's an interactive medium, like I am in front of you live. Okay, I can tell you like this is the live session, I'm talking to you. Yes, I'm seeing all your messages, all your chat. Okay, so that is the case. So that's why I want you people to promise that you will come till the very end, you will be sincere, and you will not have any problem in the class whatsoever. Okay, so that's why my request is, please don't miss the class, be sincere, come till the very end, so that we are able to get a very good result. Okay, so what is my guidelines? First is the classes are already starting now, okay, and we are starting with small sessions, not longer one. Why? Because there's a current batch going on, okay. Yes sir. And I am focused more on the, yes, making sure that the current batch pass, because their exam is on 26th, just hardly, what do you say, 20 days away, okay. So in 20 days they'll be giving their exam, so I just want them all to pass in 20 days, and you people have ample time to pass, don't worry about that, you will also pass. So let's start the guidelines. First is attend all live classes. Today I'll be doing a biochemistry revision. You all are welcome, 7 p.m. Come and attend the biochemistry revision. So attend one all live classes. So attend all the live classes. Don't miss them. Attend them. Okay. That is the first thing. Number two. Okay. Okay. Workbooks are provided. So workbook is there. So yes, write in that. And complete it. Write and complete it. Fine. So that is the second thing. So workbook, write and complete. Number three, also what else we will require from you also, that apart from that, please solve the MCQs in the Qbank. So you all have been given access to the version 5 of Victory Qbank. Version 5. And it contains around 20,000 questions of all the exam plus NEET PG, the PYQs. I'll show you the Qbank and why it is important to have the Qbank done. So I'll show you. So this is our Qbank. So I want all of you, yes, to get this Qbank done. Okay. So this is the Qbank. This one. So it will be updated to 4.5 to 5. Okay. So it contains the FMGE PYQs. All the PYQs are here. Then it also contains the TND videos. You can watch subject wise test, topic wise Qbank. Like I'll open it. So it contains all like medicine, community medicine, derma, psychiatry, surgery, physiology, pathology, OBG. Let's say I open the OBG. So in OBG, you'll have the folder like general gynecology, infections, infertility, cancers, basics of obstetric. Let's say open general gynecology. So prolapse, endometriosis, contraceptive disorder, fibroid, menstrual cycle. So let's say menstrual cycle I want to open. So it'll have 17 questions like what is menometrorrhagia, self breast examination, and the options will have all the detailed descriptions. Okay, like there's the answer and there's a question, this is the options, and this is the answer. So let me show you the answer. So this is the answer, 7 to 10 days after the menses, 7 to 10 other options. So they explain each and every option in this case. Clear? So that is the idea here. Then if you are checking the NEET PG PYQs, let me show you. So in NEET PG PYQs, you will get all the PYQs etc there. So that's what I'm telling you. You have to practice the question topic wise. Like today I'm doing general physio. You can solve the questions of general physio in that case. Okay. Like this one. Good morning, Adi. So there's a question bank like the mentor Qbank version 5. So that's why my idea is you people should solve as many questions as possible, because that will help you prepare for the upcoming exam. Okay, so practice the question and it contains almost each and everything that is there. Okay. So let's show the NEET PG question. So in the FMG Qbank, let me show you this PG ones. So let's say the NEET PG 23 paper anesthesia questions. So that is the question. Okay, what is the maneuver? The photo will be there. They'll give 4 options, and then in the edit you can see my version is looking like this because my version. So it will be like this, and the explanation will be given with details. Can you see? So this is the explanation. The head tilt, chin lift, okay, maneuver is performed. This basic life support plus other options, jaw thrust. Then this is the other ones, and they have explanation also. Morgan clinical anesthesiology version. So that's why they have all the explanation, all the finding. So that is the case. Okay. So now that will be just before the exam. Okay. Why? Because I need to go through all the important points, discuss them, revise them, and then I'll go to that part. So that's why I want you people to keep attending the sessions, and yes, then get this done. Okay. So I hope you all understood what I am trying to say to you, that you need to practice the question. Okay. Then give subject test after the subject is over, like physio test. Give the subject test after the class is over. And number 5 is attend TNDs, test and discussions by faculties. Number 6, yes, what else do we also see or have? Okay, right? So also we will be seeing what? We'll be seeing what? Yes, the revision sessions and chanting. So revision and chanting. So revision और chanting भी हम लोगों ने करनी है. So revision and chantings we will be doing. Okay, so revision and chantings. Okay, next question. Okay, and if you do all these steps, surely you are going to pass the exam without any problem. Then we will finish the main subjects in 3 months. So the target is like July, August, September, October. So October we will finish all 19 classes. So classes will be finished, and then we start with the revisions, the TNDs, test and discussions, then chantings, PYQs, image based sessions, and in the end I will start doing my expected exam sessions. What I do? Expected MCQ session. So that I do in the last 1.5 months before the exam, and I discuss important questions and how to approach them and how to solve all this kind of question. So that will be occurring not now but towards the end of the session. क्या बात है Swarab? Congratulations. Okay, so NEET PG, my advice is give the this August NEET PG and use your FMG notes now. Okay, just use your FMG notes. During internship, it's difficult to prepare for the exam separately. Use your NEET PG notes and prepare from that. Okay, this FMG notes and prepare the NEET PG for that. Once you've done the internship and you get free time, give fully devoted time for your NEET PG. Don't go for job, JR, medical officer, and then prepare. Once you go into any job and work, you can't study. So just like FMG, but only studying, for NEET PG also need to be just only studying. So stop everything, join some classes, maybe do self study, but prepare for PG without any distraction, and you will get. Okay. So I'll be starting the session. Let's start. So the first thing we are discussing is general and our cell physiology. Now what do you study in this general and cell physiology? Now remember, all the living things are made up of cell. So unit of life is the cell, and all the cells in the body, they are having the similar parts. Okay, and what each cell contain? So and cell has and Cell has first cell membrane, CM, and cytoplasm. And cytoplasm contain cell organelles. So it contains cell organelles. Okay. So what are the cell organelles? What are the cell? Let's start. Nucleus. What does nucleus contain? Tell me. What does nucleus contain? It contain our what structures? Yes, it contain the chromosomes which have the DNA, and DNA is then used to make the RNA and the protein. Then we have mitochondria. Yes, very good. Saranya. This is ATP formation. So where are the ATP coming from? Yes, from this cytoplasm is this. Okay, it contains the water, or is ICF, intracellular fluid. Then there's a plasma or cell membrane. I'll discuss it later. Then this nucleus, it is inside. Okay, again it play a key role in the mitosis and the division of cell. Then we have rough endoplasmic rhythm. What is the function? Rough. It contains ribosomes for protein synthesis. For protein. So protein synthesis occur here. So protein synthesis. Then we have the, yes, this smooth endoplasmic one. This endoplasmic reticulum, rough one, is it makes steroids. Steroid synthesis. So it is for steroid synthesis. So what synthesis? Steroids. Okay. And Golgi apparatus, it make phospholipids. Phospholipids, okay, synthesis. And vesicles. It make vesicles which are carrying neurotransmitters, etc. Vesicles synthesis. Okay, so let's zoom in. So this is Golgi apparatus. It contains small vesicles, and these vesicles go to the cell membrane. So in short, it makes the cell membrane. So that is why, what are the basic idea? Our nucleus is the one which contain all the DNA and the machinery for replication, protein synthesis. Then we have the mitochondria which make the ATPs. Cytoplasm contain all the water and the structures. Then we have a cell membrane which surround the cell. Then Golgi apparatus make the phospholipids which are a component of cell membrane, and also made small vesicles. I'll show you here. See. So these are the vesicles. Who make the vesicle? Golgi apparatus. These vesicles go and merge with the membrane and release the content outside. These contents which are released outside, they are the neurotransmitter, hormones, growth factor. So that's why who secrete them? The answer is they are secreted by our vesicle merging with the membrane. I'll discuss it later, but right now just remember these are our main transports. Okay. Now let's discuss the cell membrane. So cell membrane is very important because it is like the house key walls. Without the wall, the house will collapse. So the best model to describe our cell membrane is our Singer-Nicolson fluid mosaic. Very good, Said Usman. Mosaic. Now what is this fluid mosaic model? So cell membrane contain lipids and proteins. So lipids and proteins. Lipids are around 45%. Proteins are 55%. So what is more in the cell membrane? Obviously the proteins are more. People think it's lipids are more. No. Proteins are more. Now question is, what are the main lipids? So question is, what are the main lipids? So remember, the main lipids are phospholipids. Okay, and phospholipid are having two ends. So it is having two ends, head and tail. Head and tails. So what are the two ends? They are the heads and the tails. Okay. Now what is the funda about the head and tails is. So head and tails is what? First, polar head. So what is the first thing? First is there are polar heads. They are made by phosphate group. Made by phosphate group. Phosphate group. Okay, like this. And they love water. Why? Because they have a charge. So due to the charge, they love the water. They love water. They love water. So what is it called as? Hydrophilic. Hydrophilic. So remember, anything which is charged is water soluble and love the water. Okay, so remember, water dissolve all the ions, all the salt. Why? Because water love charges. So water love charges. So remember, it is polar. Polar means it has a charge, and charge is love the water. So remember, charge love the water. क्या बोलते हैं इसको? Hydrophilic. क्या बोलोगे? Hydrophilic. So please remember, anything which is having a charge ion, it is water soluble. Non-polar tails are also there. So non-polar tails, they are made by lipid chains. And water and fat mix or don't mix? So they are made by lipid chains. So water and the yes, what do you say? Lipids, fats, mix or don't mix? Don't mix. So that's why they hate water. So they hate water. So what they are called as? Hydrophobic. Hydrophobic. Phobic means fear. So they fear or run away from the water. Hydro. Yes. Yes, Dr. Blocks will do that. Don't worry. So it will be running away. Okay. Clear? Already expected series is going on. Okay, you can watch that. So this is the case. So what is made by lipid chains? Non-polar tails are made by the lipid chains, and they hate the, what they hate? They hate the water. What is it called as? It is called as hydrophobic. What is it called as? It is called as our hydrophobic. Hydrophobic. Are you clear? Clear? So hydrophobic is the case. Okay. Now, phospholipids are arranged as what? As bilayer with proteins inserted in between. So what are they arranged as? They are arranged as a bilayer with what inserted in between the protein. This is called as the fluid mosaic model. So what is the lipids? They are like the fluid. They are like the fluid because they are very water-like. So membranes are like very flexible thanks to the lipid. Remember oil, cream. So in the milk on the top there's a cream. Cream कैसी होती है? Very loose, very liquid-like. So that's why fluids are phospholipids, and they are arranged as bilayer with protein inserted in between. And remember, heads are outside and tails are inside. So remember, heads are outside and tails in between them, and tails in between them. Are you clear? So heads now, head. So let's see that. So you can see the head is here. So head is here, this one. So head is outside, head is outside. So again there are head and head. These are heads. But the tails, the yellow ones are the tails. They are situated where? They are in between. So these are the tails. They are between heads. So I hope you can see. So this is your Singer-Nicolson fluid mosaic model in which heads are outside and tails are in between them. So what is in between them? Tails are in between them. So tails. Now also there is what? The proteins. So proteins are present in the membrane. Okay. And the proteins are of two types. One is the protein which is at one end. So I'll write here. So right here. Two type of proteins. Two type of proteins. First is peripheral. वो बाहर की तरफ होती है, peripheral. Like this is peripheral. So I'll use the color purple, blue color. So this is peripheral. Peripheral. So this one is peripheral one. Yes, peripheral is at one of the ends. Okay, so outer side. So peripheral, and the other is our transmembrane. Trans का मतलब ही क्या है? Across the membrane. So they are across the cell membrane. So it's transmembrane or integral protein. Basically, they are from one end to other end. Like this is integral. This is integral protein. So what they do? They cross the membrane from one side to other. So they make a hole across the membrane. जैसे मान लो ये आपका membrane है. If I make a hole, so it become like this. This is the transmembrane protein. So it is inserted in the membrane. It creates a tunnel through which the water soluble things can go from this side to this side. Can you see? So this hole like thing is called as what? Is called as our, yes, integral protein. And what they create? They form the, yes, the channel or the transporter for the substances to cross the membrane. So what they do? Help. So they act as channel or transporter. So I'll discuss it later how. But remember, who make the channel or transporter? It is the transmembrane protein. Why? Because it is present from one side to the other side. So it connect the ICF with ECF. So that's why, yes, anything which is present on the outside can come inside, and the inside things can go outside. So that is why what is connecting our ICF with the ECF and vice versa? The answer is transmembrane proteins. Okay. So transmembrane proteins, what they do? They connect our ICF with the ECF. Thank you, Venkat. So they are connecting the ICF with the ECF. I'll discuss, don't worry. ये भी आने वाला अगला point. देखो. So first is, what are the functions of the lipids? So lipids in the cell membrane, they provide integrity. What's integrity means? They keep everything together. So keep everything together. So otherwise the membrane will rupture easily. So they provide strength, keep everything together. Then they are very flexible. Like this is the membrane. They're very flexible. So membranes are not rigid. They don't rupture easily. They are very flexible. So what is the word? Flexibility. Flexibility is also called as fluidity. So membranes are like a gel. Gel, like very soft, very flexible. That's why can a cell divide? Yes. Why? Because the membranes can, yes, they can pinch and separate into two. So that is why, and it is due to because of. So flexibility or fluidity is because of what? It is because of what? It is because of what? It is because of, yes, our cholesterol and unsaturated fatty acids. Unsaturated fatty acids. So unsaturated fatty acids. Okay. Then comes solubility. Now, what do you mean by solubility? Solubility means dissolve. So lipid soluble things can easily cross lipid membrane. Can easily cross cell membrane, and they can come in and out. So they can come in the cell, in and out of cell easily. Example, gases. So can the gases need any help? No. So oxygen, carbon dioxide, even what? Ammonia, N2O. Can they cross the membrane? Yes. Yes, they can cross. Thank you, Radha Krishnan. All our regular batch students are also here. So this will be the case. So they can easily cross the membrane. What they can do? Easily cross. So gases. Apart from gases, what do we have? We have fat, soluble, hormones, and vitamins. Which are the vitamins? Tell me. Vitamins A, D, E, K. So A, D, E, K. So which are the fat soluble vitamins? They are A and D and K. So what they are? A, D and K. A, D, E, K. Yes. So what they are? They are A, D, E, K. and also fat soluble hormones. Fat soluble hormones. Very good, Syed Usman. Very good, Saranya. Yes, fat soluble hormones. Which are the fat soluble hormones? Anyone? Which are the hormones? Okay, one is thyroid. Any other? Any other hormone you people know? Give the answer. Give the answer, Syed, Saranya, बाकी लोग. Yes, steroids. Very good, Syed Usman. Okay, so it is steroids. Yes, Prasanna, you are right. It is the steroid, sex steroid. Okay. Yes. Yes, it is sex steroids, like steroids and thyroid. Now understand, okay? Water soluble substances cannot cross the lipid cell membrane. Come to next page. So right. See, water soluble substances like ions, sugars, they can't cross lipid cell membrane. Can't cross lipid cell membrane. So who will help them? Yes, you are right, Dr. Radhakrishnan. Yes, Saranya. So who will help them? Okay. So obviously, what the proteins do? So proteins of cell membrane help water soluble cross the membrane and act inside. So who will help them? Yes, the water soluble substances are going to help them cross the cell membrane and act inside. And act inside. Are you clear? ठीक है. Okay, very good. So that is why the proteins of cell membrane, the integral ones, they help the water soluble cross the cell membrane and act inside. So that means who need them? The answer is this. Example, ions. Ions like sodium, chloride, calcium, etc. use ion channels. Use ion channels or pumps to cross the cell membrane. सही है. So they use, ठीक है. Whereas solutes like sugar, amino acid, urea, they use carriers, also called transporters. Example, glute. What is glute? Glucose transporter. So what is it called as? Glucose transporter. So what is it called as? Glucose transporter. So let me explain it to you. Okay. So also the water soluble hormone and neurotransmitter, like example, insulin, then GABA, glutamate. So these GABA, they use what? They use receptors for their action. They use receptor for action. So what they use? They use receptor for the action. So what the receptor do? Action. So क्या करता है वो? Action कराएगा cell के अंदर. So let me give a summary. Yes, it is glucose transport. So let me give a summary and then we will come to here. Okay. So let's start with the summary. So summary is, this is our cell. This is the cell membrane. This is cytoplasm. Okay. Now question. Let's say this is our sodium and this is our oxygen and this is our thyroid and this is our glucose. Now can you people tell me who can easily enter the membrane through the lipid part? Tell me. Who can enter the membrane? Tell me. Who will easily go through the membrane? Anyone? Very good. So thyroid can easily enter, oxygen can enter. Why? It's a gas and it's fat soluble. So they can enter the cell easily. Easy? Very good, Prasanna. Dr. Saranya, Santosh. Very easy. Okay. Yes, Sayed, it will enter easily without any problem. But the sodium, it is not allowed. क्या बोलते हैं? You are not allowed. You cannot come, get out, you bloody fool. Right? So they are not allowed. Okay. Like they do for FMG. So it's like a FMG student. They don't let you come to India and practice in India. So you want to do India practice, India job, India, they will say no, no. So what is this thing is? This is our NMC. So क्या बोलता है NMC? You are not allowed. So आप बोलते हो sir, please find some solution, sir. Okay, if you people can give the exam and pass through this exam, तो ये हो गया आपका सोडियम चैनल. So this sodium channel is like the FMG exam. Is like the FMG exam. So now can the sodium come? Yes, but only by passing through the FMG exam. And now sodium can enter. Now FMGs can practice in the country. Have you understood the story? So that is why water soluble things or the very like difficult to cross things, they need to use some kind of mechanism. Have you all understood? Same is with glucose. So glucose is what? It is also like what? Water soluble. So it also use a transporter. So again we create a transporter, and this transporter is called as what? This is called as glucose. And again this glucose is allowed entry inside. So I hope everyone understood the story. Is it clear to all of you or not? Okay. Now come to the second story. So second story is, what if there is a substance which is, yes, coming in the cell? Yes. Now what about the, yes, other things? So I hope you are understanding the class, all of you. Class is understandable. Give the answer. I'll just get my water. I forget my water for 1 second. Okay. Very good. I'm very happy. Okay, very good. Prasanna, Prakash, Saranya, Santosh. Very good, very good. Syed Usman, very good. Okay, so let's continue and let's move to the next point. So what is the next point of the topic? So audio, video, class quality, then content, like you have got the Qbank, you have got all the last session videos. So whenever you have time, you can watch the last session videos. They are all available. So last session, all 19 subjects are available in the app. Last session, 19 subject videos. So that's why don't waste time. Like, अरे sir, आज class नहीं हो गई, खत्म हो गई. So class is okay. Okay, I'll discuss Venkat that later. Okay, because we are doing it for the students who are fresh and giving the December exam. Okay, clear? So I'm focusing more on the, yes, classes for that. Okay, for you I have evening session, biochemistry, that I discuss. Okay. So 19 subjects. What is there? 19 subjects. Okay, videos are there, and you can watch them so that you are not lacking in any preparation. So I request you, watch the physiology as much as possible. Plus you will attend live with me. That will give you another chance to interact and discuss with me. So my request is do that. Okay. Ciao. So I'll come once. Okay, I'm back. Just got my coffee. Yes. And chocolates for all of you. Bucharest. Okay, so all of you can have this chocolate. So those who come to Delhi after their exam, I will give them chocolates for all of you. Let's start. So we are now continuing with the class. Now water. So proteins of cell membrane also act as antigens. Which antigens? Self antigens. Self. And other cells like and bacteria cell proteins would act as foreign antigens and killed by our immunity, killed by WBCs. Are you clear? To understand, the proteins of my cell are my antigens, self antigen, and the bacteria cell proteins would act as what? Foreign antigen. So like if there is some foreigner in India, what will the police say? Give your ID card. Dikhao bhai, dikhao ID card. Now, obviously, this terrorist from nearby countries, they don't have the Aadhaar card. So they will not show and the Indian police will arrest or maybe thai. So that is mean you have to have a identity card. So who's acting as an ID card? So remember the proteins of the cell membrane, they are acting as what? They are acting as the antigens. What are they acting as? They are acting as antigens. What they act as? Antigens, and that is why we can differentiate your cell from the outside cell. Okay, so who act as antigens? Yes, the proteins act as antigen. कौन से वाले? Self. And what are the other proteins? Bacteria cell protein. They are foreign antigens. And can you make antibody against them? Yes, and kill by WBC. So that is the story. Okay. Now come water. So what is the story about the water? So story about the, yes, example I'll discuss later like MSC, HLD. Okay. So that is the case. Okay. Example, यहाँ लिख लो. Example MHC or HLA. What is MSC? Major histocompatibility complex. Major histocompatibility complex. Just focus, Aman, just focus on the PYQs. Just focus on the PYQs. Don't try to solve all the Qbank. There are so many questions which are not even relevant to the current exam pattern. Okay, rarely these kind of questions will come. Focus more on the, yes, your FMGE and NEET PG PYQs. Okay, histocompatibility complex. So which complex? Major histocompatibility complex. Which complex? Major histocompatibility complex is the case. I hope it's clear. Sure. Next is next question. HL is human leukocyte antigen. So they are foreign or your antigen? They are which antigens, bachcha log? They are your self antigen. They are your self. So that's why my antigens, I am my antigen. Your antigen are your antigen. If you give your kidney to me, your MSC, your protein will be foreigner for me. So my immune system, WBC, will attack that and destroy. That is by organ rejection occur. What occurs? Organ rejection occur. Are you clear? Understanding? Okay. Now we'll discuss water. So water do what? Let's discuss. So basically, water use aquaporin. Water use aquaporin, which is water channel. What is aquaporin? Water channel to enter cell. So water cannot cross lipid membrane. So it use the, yes, aquaporin. Now, if something is very huge, very big, How will it cross the membrane? It uses a special kind of process called as endocytosis, exocytosis. Very good. So big substances, big substances like like proteins, hormones, bacteria, etc. cannot cross lipid cell membrane. Yes, okay. Cannot enter via transporters or channels because they are very big. So they cannot use the transporter or channels. So what they do? So they use vesicular transport. So which transport? Vesicular transport. What happens in vesicular transport? Basically, okay, the substance is surrounded by phospholipid layer to form a cycle, and then it will enter or leave. So it will enter or leave. So what is the solution? I'll discuss. So let's discuss first. Okay. So let's say a bacteria comes. This is a bacteria, and your WBC want to eat the bacteria. So obviously it's a very big bacteria. You cannot make such a big hole as the cell will die. So what do? You surround that bacteria and then you eat it. That is called as phagocytosis. And now the vesicle or the vacuole is made by the cell membrane. So cell membrane surround the object and make the bacteria and bring it in. It is called as endocytosis. Example first, endocytosis. Okay, so it basically what? Bring substance inside cell after cell membrane. Yes, after cell membrane surround it to make vacuum. Example first, example phagocytosis. Phagocytosis. So this is the first example A. So here we will, yes, kill the bacteria. So phagocytosis, yes, it is by bacteria. So here example WBC eat bacteria. So what happens? This is WBC membrane. It surround the bacteria and bring the bacteria in and then eat and destroy the bacteria. Very good. Very good, Sayyid Usman. Then pinocytosis is cell drinking. So it is cell drinking. Cell drink water. So cell can drink lots of water from the ECF, and that is called as pino or pinocytosis. What the word pino means? Drink. Pino means drink. So pino means drink. And last is our receptor mediated. It is basically for very special protein and it is via clathrin. So receptor is clathrin. So clathrin is the protein which help in bringing the receptor mediated endocytosis. Are you clear? So clathrin protein help in the receptor mediated. So let's revise. What is bacteria? Big things come. Phago. What is water come? It is drinking. Phago means eat. And what is receptor mediated spatial substance entry? So receptor mediated spatial. See, all the box and the stars are coming. No, only stars are coming. This is called as receptor. Only specific. So it is for specific substance. Specific molecule. So it is for specific substance. So specific molecule is receptor mediated. And what is the protein which help? Clathrin protein help in that. So clathrin is the coating protein. So which protein? Coat protein. It make the coating of the vesicle and help in the transport. Now if you want to throw a substance out, what do we use? We use exocytosis. So in exocytosis, Golgi apparatus make a vesicle around the neurotransmitter or hormone, and then this vesicle go and merge with the cell membrane and release the substance outside. This is called as our exocytosis. So exo is for secretion. So right here. Number 2, exocytosis help in secretion of hormones, proteins, etc. Okay. Golgi apparatus make the vesicles. Make the vesicles, and if calcium help, called as regulated secretion, called as regulated secretion. What is it called as? Regulated. How are you, Niharika? क्या बात है? Best of luck to you. Congratulations for your NEET PG exam. Just do well, don't take stress, and I'll be very happy to meet you again. Okay, with a very great PG seat. Okay, ठीक है बच्चा. Best of luck once again. So regulated secretion. So this is calcium. So this is calcium. So if calcium is causing the secretion, it is called as regulated. And if no calcium, it is called as constitutive. What is constitutive? No calcium required. No calcium needed. So what need the calcium? Regulated. And without the calcium, obviously, regulated signal is calcium. Non-regulated, no calcium. Are you clear? So no calcium is the one. Okay. So I hope it's clear now. So is this all clear to all of you? Yes or no? Understandable? Yes or no? Okay. Let me now revise and summarize. देखो, if big substance, what we use? Vesicles. And what are the two direction? Entry or exit. If entry, called as endocytosis. If exit, what is exit? If exit, it is exocytosis. So entry in the cell is called as endocytosis. Exit is called as exo. Exocytosis help in secretion. Help in secretion out of the cell, proteins, hormones, neurotransmitters, etc. Okay. So now it is two types. What are the two types? If need calcium, called as if need calcium, called as regulated. Why? Because calcium will control. So called as regulated secretion as constitutive. Okay. So without calcium means you don't need any control. It's continuous. So continuous is like a constitutive, and with calcium, like only when the calcium comes, let's say neurotransmitter, only when I give a stimulus, like I say hello, your brain release calcium and calcium release the neurotransmitter and you hear my voice. That is called as regulated secretion. Okay. Now endocytosis is, yes, entry inside the cell. Thank you, Srijan. Thank you very much. So our current batch students are still very, what do you say, involved and attached, because daily we are having sessions for them. we are having revisions, we are having question discussion, and this is our mission to make all of them pass. And that's what I keep telling you. Okay, we focus more on the, yes, students. We are not for like, students are here, we don't give a damn. Okay, हो गया, पैसा दे दिया, वीडियो देखो, तुम्हारी मर्जी, ठीक है, you want to watch video, watch, else we are not, give us the money. We are not, we keep a track of each and every student, we talk to them, we discuss with them, we ask their problem, we solve their queries, we have telegram groups for continuous interaction, we have regular session, almost daily I am taking session with them. I've taken 18 session till now. Okay, 15 plus 3 additional one, so 18 sessions I had in the evening in the past 1 month, and I was having all kind of important topic, almost 7-8 subject we have finished, and we discuss the question, and I know the results are going to be fantabulous for the NLC student. Any student in NLC, they never regret that they came here. Reason? They get everything which is required. Only thing is, you just need to, like, you know, you're buying a thali. This thali contain everything: raita, salad, dal, or if you're non-vegetarian, it contains that stuff. So, okay, roti, chawal, okay, rasgulla, gulab jamun. Only thing is, you need to eat it and not, you know, waste it. So once you eat it, you are going to. Okay, thank you, Venkat Sai Thiwa. Chal. So it is basically, yes, it is for, yes, so endocytosis. So basically what occurs, so it is 3 types. First is cell eating. What is cell eating called? It is, thank you, children. It is called as phagocytosis. Phagocytosis. So what is the first one? Phagocytosis. If cell want to drink lots of water, so do cell drinking. Pinocytosis. Very good. Pinocytosis. And number 3 is our, yes, what is the third process? Third process is specific substance. Specific entries. Specific. So what is it called as? It is called as receptor mediated endocytosis. So only when it can bind to the receptor, it can. So receptor mediated endocytosis. What is it called as? Receptor mediated, mediated endocytosis. Okay, and who help? Helped by clathrin. Clathrin protein. So who help in that? Clathrin protein helped in that. Okay. Clathrin. Very good, Saranya, Said Usman. Very good. You all are rock stars. So you people will do very well if you continue studying. Very good for the exam. At the end, what matters is your hard work and sincerity. Okay. If you're sincere, hardworking, you're going to pass the exam. Else, it will be difficult. Okay, let me show you. Let's say this is our nucleus and this is our WBC. So this is our WBC, okay, and it's see a bacteria. ऐसे ये आ रहा बैक्टीरिया घूमता फिरता, तो ये आ गया बैक्टीरिया. सो बैक्टीरिया कोई बनाते हैं अच्छा सा, ये हो गया बैक्टीरिया. ठीक है, सो ये बैक्टीरिया आ गया और WBC को दिख गया, तो क्या बोलता है WBC लाइक पुलिसमैन. और ये क्या आपका, ये हो गया क्रिमिनल. तो क्या बोलता है WBC, चल भाई आईडी कार्ड दिखा. ओके, शो मी योर आईडी कार्ड. तो बैक्टीरिया डजंट हैव अ आईडी कार्ड, सो ट्राई टू रन. सो पुलिस come near the bacteria. As it come near the bacteria want to run away, but the police surround the bacteria. The police will surround the bacteria like this. Okay, so bacteria will be surrounded. So our cell make a depression, and as the cell make a depression, it go near this. So now police is surrounding the criminal from all sides. Remember, policemen capture, and now the policemen will not stop at this. They will keep on surrounding this terrorist. Clear? So they will keep on surrounding this terrorist. Okay, and eventually the bacteria will be surrounded from one of all the sides. So from all the side, the police will surround the bacteria. And now there is no option but for the bacteria to surrender. And now the police comes and police surrounding. Can you see this? And eventually what will occur? These two ends meet. As these two ends meet, they become one. Can you see this? They become one. Can you see this? They become one. And you just need to break the middle part. As you break, this is a free floating vesicle. And this vesicle come inside the cell. And here you have arrested this criminal. And now, okay, show me ID. Okay, not. Oh, you are from that. Terrorist, different. And you will beat the terrorists very bad. And you beat them, break them, and eventually you keep on eating their proteins by enzyme. And one day the bacteria is dead. The bacteria is gone. And that is how you kill the bacteria. I hope all of you understood this process of phagocytosis, how the cells kill the bacteria. Okay, so let's continue. Let's move to the next topic. Okay, so let's see this. Just 1 minute, okay. Chalo, okay, let's continue. Now we'll discuss body fluids. What we're discussing? Body fluids, okay. So what is the story? So remember, 60 to 70% of all your body weight is water. It is called as the water, but that is in adults. In adults. But remember, in newborn, 80%, 75 to 80% is total body water. Okay. So what is the total body water? 75 to 80. And what are the two parts? The two parts are ICF and ECF. Is ICF intracellular fluid? And ECF extracellular fluid. Now add an extra point. So they are the ICF and ECF. Very good, sir. So I am good, Dr. Hari Rio. I am good. Now, so ICF is inside cells. This is outside cells. Thank you, Dr. Detoxin. Okay, so this is the story. Now let's start. Okay, now we will discuss something very essential. So these are the two parts. Okay, first, now remember, a newborn has ECF 40% more than ICF, which is 35%. This is not in the last year notes. I'll tell you, I update all my notes. These are the current latest notes. It was not in the last session notes. So if you're reading last session, you might miss. Okay, newborn is, and at 3 years of age, the ECF become equal to ICF. So when does the ECF become equal? The answer is it become equal. Okay, say it, understand. तभी मैं कह रहा हूँ, एक्सप्लेन कर रहा हूँ। ये जनवरी 2024 का एग्जाम 25 का एग्जाम क्वेश्चन था, ये था जैन 2025 FMG exam question. And I know all the PYQs, almost all 19 subject. That's why I'm the one who do expected question or discussion of the important topics. Okay. Fine. Now, next question. What are the other changes that we see? So, other changes that we see is in adults, the ICF become more than ECF. Almost double. Okay. How? Let me write here. So what happens is basically the in adults, out of total body water, 2/3 is ICF and 1/3 is ECF. 1/3 is ECF. Okay, 1/3 is ECF. Now ECF has two parts. ECF has two parts. So I'll make a diagram here and then explain. So make a diagram with me. So these are our cells. These are the cells. So these are cells. They have is ICF. Then this red color structure you see is the blood vessel. It contains plasma. And this thing here is IF. These two combined is called as ECF. So what is the story? Let me now explain. So understand. So basically what happens, the ECF contain two liquids. So ECF contain 25% plasma where? Inside blood vessels. So this is plasma and 75% is IF, interstitial fluid. So what is interstitial fluid? Let me now give all the ideas. So plasma is inside the vessel. So plasma is where? Inside the vessel. So please don't forget this. So plasma is inside our vessels. Okay, inside the vessels. Okay. Then what is ICF? ICF is our inside the cells. This is ICF. Is your ICF. Okay. And this liquid in between the two chambers, this liquid is the interstitial. Now this interstitial fluid is also called as the third space. What is it also called as? Third space. Why? Why it is called as third space? Anyone? Why it is called as the third space? कोई बताएगा? Anyone would like to put some emphasis? Why the third space? Reason? Anyone? This is the first space. This is the second space. This is the third space. So what is the third space? It is between the cells and the plasma. Okay. So what is the location? Why? As it is between cells and vessels. So that's why it's called as third space. Okay. Third space means the third place. And it is very important. Why? Because edema etc. occur here. Number two, also called as lymph. Also called as tissue fluid, and it help form lymph. Help form lymph. So what is lymph made from? Lymph is made from our which liquid? Interstitial fluid. Okay, so interstitial fluid or the tissue fluid help form the lymph. Okay, And lastly, it is same as plasma. It is same as plasma, but no plasma protein. No plasma proteins. No. So where are the proteins present? Only in the plasma. So what is missing? Plasma proteins are missing from the interstitium. Okay. Now remember what happens next. The glucose which is present in your plasma will go to what first? It goes to interstitium first, and from interstitium it goes to all the cells. So that means who will give glucose, oxygen, nutrition to all the cells? Plasma, but directly or via interstitium? Via the interstitium. So how does everything which is present in your blood reaches the cells? Answer is through the interstitium. So remember, all the cells of your body, they are connected to each other through the plasma and the interstitium. Have you understood this point? Yes or no? So where does all the exchanges occurring? Is the interstitium. So cells get all the glucose, all the oxygen, all the nutrients directly? No. They get it through the interstitium. Interstitium is like the, क्या बोलते हैं आपका? Is like the area where you just order, hey, give me this, you get this, okay? And what is the source? Is plasma. So you can say plasma is like, let's say our Flipkart or what, क्या बोलते हैं ना, this Blinkit, and you order and now you get the supply. So that is why it is a transfer medium in which everything comes. Okay, done. So where will bacteria want to come? A question for you people. Where does bacteria want to come? Bacteria को कौन सा area पसंद है? Cells, plasma or interstitium? So remember, bacteria want to come to which area? Bacteria को कहाँ पे आके मजा आ जाएगा? Tell me bacteria. These are new notes. Tell me. So where will bacteria will go? Obviously, tell me, Syed, Dr. Saranya, Shrijan, okay? Those who are in the class, can you give the answer? Aman, give the answer. बताओ. So, okay, see, understand. The bacteria will enter the interstitium. Okay, why? The answer is very simple. So let me explain you some pathology concepts here. Understand. This is, let's say, your skin. This is your skin and it is made up of cells. What is it made up of? Epithelial cells. What is skin made up of? Cells. So these are the cells. And this is our interstitium. This is the interstitium, IF, and it contains glucose, oxygen, amino acids, and this is our blood vessels. This is blood vessel. Blood vessel contain the WBC. So WBC are present here. So now bacteria will not enter the blood. So if there is an injury, I'm shaving, I'm eating, can the cells of epithelium, skin or oral cavity be damaged? Yes. So while I eat, this cell got damaged. So cell will get ruptured. It create an entry and now ये साले हरामी बैक्टीरिया लोग घुस आएंगे बॉडी में. कौन से? ग्राम पॉजिटिव बैक्टीरिया. So these are gram positive bacteria. Just say bacteria. So now the bacteria enter what space? They enter interstitium. So please understand, bacteria enter the interstitium. So where do bacteria go? And now they get glucose. मजा आ गया. Like terrorists or refugees from other country want to come where? To India. So every year lots of this come to India or vice versa, or they go to America through the donkey route. Remember? So now they cross the border and they come to the interstitium. और क्या बोल रही है? ग्लूकोज मजा आ गया, ये जिंदगी बन गई. And now they are increasing in number. and that why it cause some symptom. So WBC क्या बोलेगा, अबे यार बैक्टीरिया आ गया. So police will go and investigate the matter. So your WBC will enter what, the interstitium, and क्या बोलेगा, अबे कौन, ID card, they don't have the ID card. So bacteria will, yes, not show, and the WBC will kill all these bacteria. So क्या करेगा WBC, इन सबको खा जाएगा, and this area become free, and this epithelial cell are regenerated, new cells are formed, and that is how infection is over. Okay, some bacteria might enter to lymph, which causes the lymph node to become enlarged. That is why lymph node of the area where bacteria come can increase in size. So have you understood it now, कि why lymph node become big? Because you don't want the bacteria to spread everywhere. So that's why to prevent the spread, we need to increase the size of lymph node. So have you understood why lymph node become big? If there's an infection, because there will be lots of bacteria, and the bacteria can go to every part of body. So you will make the roadblock. क्या बोलोगे? A lymph node. Make sure no lymphatic can go to other area without checking. So that is lymph node occurs. So क्या हो जाएगा? इस area का lymph node बड़ा हो जाएगा to stop the interstitium from going to the other area interstitium. Have you understood? This is lymph node. What lymph node will do? Not let the bacteria go to the other interstitium. Are you all able to understand? So that is the purpose of lymphadenopathy. क्या बोलते हैं इसको? Lymphadenopathy. Okay. Thank you, Dr. Saurav. Thank you. Best of luck for your NEET PG exam. And see, that is the best thing I get. I told you, night I had class since 8:30 a.m. in NEET PG till 9 p.m. So I took class till 9, and then after 9 I came and I took a online batch our class of tha till 10:30. So it's almost 14 hours, and I said the only thing I do which make me happy is the messages of students who pass the exam, who were able to clear their session and become doctor, able to work in this country, earn money, help their parent. I think that is the best thing you can do. Clear? Giving someone a small chapati, banana, food is not good. Reason? You give him food one day, they will eat next day. Okay, ना कई लोग के धर्म के लिए, क्या charity के लिए, they will donate food in the temples और चर्च मस्जिद. क्या बोलते हैं, आज हमने सबको खिलाया, कुर्बानी का हमने सबको बांट दिया खाना ईद का. That is one day. But by making the students pass, they become self-sufficient, and they can now work all life and support so many people, heal so many disease. So that is why I think give someone a fish, they will eat for a day. Teach them fishing, they will eat all life. That is what I believe in. Making people pass and work as doctors, and obviously that is the purpose I have. So I really want you people to pass, and not just facts कि ऐसा होता है. No, I want you to understand. Biggest thing is in NLC we focus more on understanding of the concept, not just plus updated material. We just don't keep teaching same. Some places they will teach 12-year 13-year-old notes, and you can even see that sometimes dates 2013 date delivery. Arey Baba, why are you teaching such old notes? Reason? They don't want to make efforts. Okay, for me 14 hours is average work. You can ask any student who are currently studying. I daily work 14. After this class I have an offline session for new batch plus the offline batch. Then night I'll come back and take an online class till 9:30. So that's why I am working hard, and I want everyone to pass. So sincerity should be two-way. If I am working hard, I want you people to also work hard. So I hope everything is clear to all of you. कि bacteria and infection, why WBC need to go to the interstitium. So where does inflammation occur? In the interstitium. Where is the bacteria coming? Interstitium. Where will all the pus be formed? So this pus form, you can see the lines of the sear will be bulging here, and there is pus under the skin. Why? Because the bacteria are accumulating and WBC are killing them, which cause formation of pus. So pus is there, we need to drain the pus. By making incision, pus comes out. Are you understanding? Okay. So now let's continue. We'll discuss the question estimation of body fluid chambers. So how will you measure them? We use a dye dilution method. What is that? We inject dye in the chamber. What we do? Inject dye in chamber. Then dye spread everywhere in chamber. Everywhere equally. And we take sample of dye from chamber and this gives dye concentration. So we take small, yes, amount sample of dye from the chamber, give dye. So what is the volume given by? So volume of chamber is given by amount of dye divided by the concentration of dye. So amount of dye divided by what? By the concentration of the dye. Are you clear? Yes. So very good, Saranya. Yes. So amount of dye by concentration give you the volume of the chamber. So now what are the methods for this? So for total body water, we will use water. But we use heavy water. So we use heavy water, which is made from what? Which is made from what? It is made from, yes, isotopes of hydrogen, called as what? Called as deuterium or tritium, called as D2 or T2. So that is also called as heavy water. So what is D2O? Deuterium oxide. T2O is tritium oxide. So what is the dye we use? D2O, which is deuterium oxide for total body water. So remember, water is given by water. Very good, Saranya, Saeed Usman. Yes, water we measure with water. So water with water. Water with water. For ECF, we want a dye. Okay, blur. Venkat, let me check. 1 second. Okay, it's actually the capture is automatic. So sometimes what it does, it fluctuates. Now it's clear. Okay, Venkat, it's okay. Venkat is always very, like his sharp eyes. Very good observation. Yeah. So we want the dye to what? Yeah. Thank you, Venkat. So dye should not enter the cell. So dye should not enter the cell. And stay only in ECF. So that means it should be only in the, that means water soluble. Big example, we can use inulin. कैसे याद करेंगे? ECF, E, inulin. So ECF is inulin. So ECF is given by what? Is given by inulin. So ECF is given by inulin. Then we can use our sucrose or mannitol. None of them, they are actually water soluble and none of them can enter the cell. So they stay here. They stay outside. So they stay outside. Now plasma. For plasma, the dye should stay in the plasma. So dye should stay here. It should stay in the plasma. So what is that? So we can use plasma protein. Example, we can use albumin, but should be combined with some isotope. So radio isotop labeled albumin. So this albumin cannot enter the cell. It will stay outside. Or Evans blue dye. Evans blue dye. So what dye? Evansky blue dye. So which dye? Evans blue dye. So which dye? Evans blue dye. Okay. Now for number 4, ICF. We cannot put dye in the cells. Okay. Yeah, we can use iodine I-125 or 131. So we can put iodine as a isotope. So iodine can be put as isotope. Yes, say you write 131 or 125, 123. But again, understand. So ICF, we can't put dye in the cells. Cells are very small and lots of cells are there. So we do indirect total body water minus ECF. So that's why it's given by 2 dyes. Which 2 dyes? D2 and inulin. So D2 for total body water and inulin ECF. Inulin for ECF. Okay, now let's see. Let's do some question. Are you ready? Are you ready for questions? Okay. Let's first revise total body water. What are the 2 divisions? ICF, ECF. ECF 2 divisions, IF, plasma. Total body water is 60%. ICF 2/3. ECF 1/3. IF 25%. Plasma 75%. Are you understanding the maths? Sorry. My mistake. So IF is this, plasma is 25. Are you understanding? Very good. Now I will ask you a question. What dye we use for the total body water? Which dye is used? Tell me. What is the dye used for total body water? Tell me. What dye you will use? Give the name. Very good. D2, deuterium oxide. Very good. Shabash Shahid. Abu Bakar Ahmad. Very good. So Abu Bakar, I think it's Abukar. Abukar. Venkat Sai Siva, Saranya, Santosh. Very good. It's D2. For the ECF, what is the dye? ECF. ECF, yeah, we can use T2 also. For ECF, what we use? ECF. Give the answer for ECF. And even what is ECF? Yes, we use inulin. So inulin is used for ECF. So inulin will be used for the ECF. So what is inulin used for? It is used for the ECF estimation. Are you clear? So ECF estimation, inulin. And then come plasma. What we use for plasma? Yes, very good. Venkat, Syed, Abu, Kar, Saranya. Very good. We use the Ivan blue or albumin. So now, is it clear to all of you? Yes. Very good. I'm very proud of you all. Now let's continue. Now we'll discuss the topic called as transport across our what? Cell membrane. So what we're discussing now? Transport across cell membrane. So remember, there are two kind of transport which can occur. What are they? They are active and passive transports. What they are? Active transport and the passive transport. Now what do you mean by active and passive? Let me explain. So active transport basically means what? First understand, active is against gradient. Active is against the gradient. So the active is against the gradient. See, iodine radioactive, Sayed, is a isotope. We combine it with the albumin to get it. So yes, it is radio labeled now. Normal albumin is not useful. We use radio isotope labeled albumin. So हर चीज लिखने की जरूरत नहीं है ना. When you revise, you don't have to write everything. Otherwise, it will be just cumbersome. See, I teach you how to revise. Take a empty pen paper and on the paper write the main words. Don't try to put ICF is equal to the, no need. Just write ECF and 1/3. That's it. Okay. So need to explain the things when you are making a revision very simplified. See, this is simple summary. This is your detailed description. Do you think we need to write this much to revise? This is when you read. That's why people have difficulty revision multiple time. Why? They are reading this total body water, this. Arey Baba, when you want to revise, just take and write rough points, and this will help you remember much better. Are you clear? So how do you remember it much better? The answer is this is given by this particular method. Are you clear? Okay. So what is active transport? So it is along or against gradient. Obviously, active is against gradient. So what is active is? It is against the gradient. So what is active is against gradient. What does that mean? It is that mean it is from which concentration? It is from low concentration to high concentration. That is high. That is called as uphill. Like you're going to the top of a mountain. Like you are here and you're going to a mountain. It is easy or difficult? It is very difficult. Why? Because you are below at lower level of gravity and you're going against the gravity. So that's why it is going to be uphill task. Okay. And what do you need? Energy. Need energy, ATP. So we need energy to climb the mountain, and example is pumps. So pumps are our what? They are our active transport. So pumps are what? They are active. Why? Because pumps will give you energy. So you need a motor, car, pump, or your muscles, which again use the actin myosin, like muscle pump. So you climb. Whereas passive is along gradient. Passive is, what is along gradient means? It is from high to low concentration. Called as downhill. So if you are already on the top of a mountain, you don't need energy to come down. Someone just need to push up and you fall down. So that is the case. Then this cup of tea, if it is hot, it will cool itself. The heat of the cup go to the room which is cool, and that is passive. But if you want to make it hotter, what we need? If I want to make a cup of tea which is at room temperature, what I need to do? I need to heat. I give energy from the bottom, and that is why heating a cup is an example of active, use energy, heater, and the cup getting cool in the room is an example of passive. Okay, so no energy needed because you are already in the high energy state. So no energy needed. No energy needed. Clear? So what is not needed? No energy. Very good, Sayed. And finally, example, diffusion. Not diffusion and osmosis. But remember, osmosis is only water. All others can use things, but osmosis is only water. So what is osmosis? only water is osmosis. What's osmosis? Only water. So what is osmosis? Only water. Are you clear? So osmosis is our only solely water. Are you clear? That is osmosis. Okay. Now let me ask you a question and you tell me it is active or passive. Okay. So question. Okay. Tell me active or passive. Okay. Study and forgetting. What is active? Studying or forgetting? Which is active? Which is active? बताओ बच्चों. What is active? Studying is active or forgetting is active? Give the answer. See, study is what? It is active. Why? Because you are going from low knowledge to high. Okay? So going from low knowledge to high. Correct? And what is forgetting? Easy. Don't study, and it's from whatever you study. Like we study 1 point, gone. So that's why forgetting is passive. You don't need effort. No effort needed. No efforts. Here, study daily our classes. Then we remember something. Forgetting, don't study for 1 day, all gone. So that's why studying is very active, but forgetting is passive. So that is why you need lots of effort daily to learn something. And forgetting is so easy, clear, except few things. Okay, so easy, except few things. Like what are difficult to forget? Which is difficult to forget? Yes, like bad memories because they are stuck to your brain, so they are like stuck. So bad memories. Okay, now let's continue. Now we'll discuss the types of active transport. It is a very favorite topic of examiner. So what are the types? So two types of active transport right here. So two types of active. One is primary active, other is secondary active. Now, what do you mean by primary and secondary? So in primary, there is direct use of energy. Very good, Saranya. Direct use of energy. So we require energy directly. So there's a direct use of energy in this case. Okay. In case of secondary, no energy is required. So no energy is needed. So what is the story? No energy is needed in this case. Okay, in case of what? Secondary active. So no energy is needed. And in case of primary active, we require the energy all the time. So where we need energy? In primary active, there is direct use of energy. In secondary, we don't need any energy whatsoever. So that's why energy is for primary, not for secondary. Not for secondary. Okay. Okay. Now, sorry, no energy needed active directly, but indirectly. So there is no direct energy use, there is indirect use of energy. Okay, how? Let me tell you. Okay, so direct use, example are pumps. Example are pumps. So pumps are what? Active transport. Example like proton H plus pump. What does? Already stomach contain more acid, still it pump acid out against the gradient. So what does? So what happens? Already more acid, H plus, in stomach cavity, still throw H plus out into cavity. So along or against? Against gradient. So what is it called as? It is called as which transport? It is called as our passive transport or active transport. Okay. Like a papa want to enter the bus. The bus is overcrowded. So papa start punching. Abbe hut away tumhare maa ke hut. This is primary active. Why? Papa enter the bus using the energy of his arm. That is the pump. So pump use energy to move itself in the cell. That is pump. Then what is the secondary? Here no energy is used by the secondary active. So it is dependent on the pump, coupled with symport or antiport. Example, sodium symport is coupled with sodium potassium pump. Which pump? Sodium potassium pump. I'll tell you how. So first question, sodium potassium pump is which transport? Primary active. Why? It use the ATP, and as it use the ATP, it generate energy, and this energy help in sodium movement, and sodium come alone or bring glucose, amino acid. So they are not using energy. There's no energy used for glucose, but the pump is bringing sodium, which bring the glucose. That causes the energy. So that's why energy for glucose is used indirectly, because pump is giving the energy, and that help the glucose. Like papa is standing outside the bus. Papa is punching and entering. Punch. Who's punching using energy? Pump is papa. Papa is the pump. And as the sodium come, the pump sodium, it bring glucose also. Glucose is like the son of the pump. So that's why papa comes, son come, and who give the energy? The papa give. Papa say, hey, get, my son want to come. Hato, my son. And he bring or pull the sun inside. Sun is glucose, amino acid. So they are secondary active. So remember, all sodium symports are secondary active. Why? Because they use the energy of sodium potassium pump. Example, sodium amino acid symport, sodium iodide symport, sodium glucose symport. So all are secondary active. All are. Why? Because energy is used by the pump. Who use the energy? Energy used by sodium potassium pump, not by the glucose. So glucose become dependent on the sodium. It's like your friend is coming on the bike. Whose bike? Friend. Whose bike? Friend. Whose petrol? Friend. Whose money? Friend. So friend is the pump. And you, glucose, sit at the back. You are now using no energy of your own. So lift given by you to your friend or vice versa will be secondary active. So whose money? Friend. Who's getting free? You. That is secondary active. That is why friend is primary active and you at the bike are secondary active. You don't use any petrol, any money. Will you give money to your friend? Never give money to your friend. It's his duty. Okay. That is why sometimes they say दोस्त को क्या बोलो? पापा बोल. कहते ना अबे दोस्त नहीं पापा बोल. पापा क्यों? पापा की तरह ही तो एनर्जी खर्च कर रहा है वो. So दोस्त is the papa. So that is the case. So remember the mnemonic: papa pump, Pump is primary. Secondary, sa. Symport. So symports are secondary. So pumps are primary, symports are secondary. Pumps are primary, symports are secondary. Very simple cheat code. Now, one more thing. What is this sodium glucose symport also called as? So it is also called as SGLT. What is SGLT? Sodium glucose linked transport. Sodium glucose linked transport. So sodium glucose linked transport. Very good. And it is 3 types. First, the glucose come from the food in the intestine. So SGLT1 in intestine. First time glucose comes to your body, intestines. Number 2, then it gets filtered in kidney. So SGLT2 is in kidney. How many kidneys we have? We have 2 kidneys. So kidney double. Intestine is only one intestine from oral to anal and एक ही tube जा रही है, one intestine. Parts हैं small intestine, large intestine, but it's one intestine. And then 3 is in the brain. So brain is having what? 3. कैसे याद करोगे? Brain has cortex, brainstem, spinal cord, 3 part. Then also 3 system. What are the 3 system it has? So it has 3 system. Like what? It has the central nervous system. Then it has a peripheral nervous system and autonomic nervous system. So that's why brain and muscles. Brain and muscles, they have SGLT3. So this was a question asked in the recent exam. Are you clear? Now we'll discuss passive transport. So passive transport is diffusion. Sorry. Passive transport is diffusion. Okay. Second, depends on concentration gradient, that is high to low concentration, which I discussed already. High to low. High to low. What is it called? This is called as fixed loop. Called as fixed loop. Then there are 2 types of diffusion. 2 types of diffusion. A, simple diffusion. No carrier needed. Example, oxygen diffusion. So oxygen to move doesn't need any, so no carrier needed. So that is the case. Clear? Whereas if you need a transporter something, it is called as facilitated diffusion. So what does facilitated means? You need some kind of transporter or help. That is called as facilitated one. Okay, क्या चाहिए से? Need carrier. Need transporter. Example, glute. So glute, without glute, it will not. So that's why facilitated is dependent on the transporter. So it depends on the transporter. It's called as a facilitated. It's called as a facilitated. So let me make a simple summary. Write the summary. Write the summary. So first is our primary active. Pumps. Primary active are pumps. So primary active is pumps. Okay. Then secondary active. What are they? They are symports. Symport or antiports, sometimes also called as exchangers. Again, they are secondary active. Then we have the facilitated diffusion. Here it is carriers or transporter, but not simple. And finally, simple diffusion. Simple diffusion. So what happens in simple diffusion? So what happens in the simple diffusion? So very simple is nothing. No transporter needed. That is simple. Are you clear? These two are active, so they are against gradient. These two are simple, so along. Are you clear? So this is what they will ask you. Okay. So now I'll ask you a question and I want you people to respond and explain to you. Have you understood or not? Are you ready? So I'll be discussing it and I'll be explaining to you all. Okay. So let's see that point. Okay. So are you ready? So let's do a summary and we will see the summary. So summary is something like this. Okay, understand? Summary. Let's say I give you a question. This is ICF, ACF, and the photo comes like this. Okay. I hope it's visible to all of you like this. These are some molecules. And they gave something like this. Okay, and then these molecules come, they bind here, bind here, bind here, and then they go here. So can you tell me which transporter is this? Active, passive, secondary active, primary active, what is this? Can you tell me which transport is this? Anyone? First, what is the direction? High to low, low to high. Direction. Remember, it is high concentration. It is low concentration. So it must be passive. This is passive. And since it is using some kind of carrier, it becomes facilitated diffusion. It cannot be active. Why? Because the direction is The direction is from higher to lower. So it is passive. So answer is simple or facilitated. The answer is passive. That is facilitated diffusion. Okay. Done. Have you all understood this question? This was 2023 NEET PG exam question. And I have covered all the PG exam questions in your class. You will not face any problem whatsoever here. Done? All of you agree this? Okay. Okay. Now let's come back. So is it clear to all of you? Now remember, first question. All exchange between cells and blood vessels. So all the exchange between cells and blood vessels occur in the interstitium. Occur in the interstitium. Okay, that's the third space. Okay, like oxygen come, hence it form the internal environment of body. Hello, Dr. Saab, of the body. So who make the internal environment of the body? The answer is it is made by what? Our, yes, interstitial. and called as the internal environment of the body. Okay, so like fishes, where do fishes live? Not in the air but in the water. So that water of aquarium, sea, pond become their environment. Clear? Then when you go to space, in a space station ISS, like recently our Indian astronaut went, so this will be called as what? Internal environment. So that spaceship become their internal environment. Are you clear? Okay, Saurabh, it will come, don't worry. Okay, so we are making regular videos, daily I'm taking sessions. Okay, so it will be, yes, it form the internal environment called as milieu interior. Milieu interior. So are you clear? So it is milieu interior. What is milieu? Milieu is environment and interior means inside. So just like fishes survive in water, our body cell require a water. This is called as interstitial fluid. And this was coined by, and this term was coined by father of physiology, called as Claude Bernard. Claude Bernard. Are you clear? So this is our Claude Bernard. Are you clear? So clear? So this is called as what? Claude Bernard. Claude Bernard. Okay. Fine. And remember, we have an environment. The environment is not only present, but we also need to maintain. So the composition of environment is maintained. The composition of internal environment is always maintained in a steady, constant state. Example: pH, blood pressure, heart rate, temperature, etc. remain in normal physiological range. Normal physiological range. This is called as homeostasis. This is called as क्या बात है, very good. See, I told you doctor, just do the sessions I'm doing. They will cover 70-80% of topic and questions. People say I'm trying to make this. Point is, I want to cover every important topic like congenital heart disease, neonatology. So I cover 70% of any topic like biochemistry, psychiatry, anesthesia, derma. My target is simple. I want to make sure all my students pass. And if the, yes, you can all come. Okay, if all the students are watching them, they will get all the full revision. No need to spend money on the very costly revision, 10,000, 15,000. No need. Clear? Just expected session cover all the revision. In fact, it cover everything. Those who miss the subject also, like Dharma nahi kari, I'll do. Almost except STD and cancer, I did all the Dharma sessions. Okay, so that's what I'm telling you. Okay, just have trust in my ability and your hard work, and you will clear the exam. Okay, so it's called as homeostasis. It's called as the homeostasis. What is it called as? Homeostasis, and it was coined by, it was given by Walter Bradford Campbell. So Canon gave which term? He gave the term which is now called as homeostasis. Okay, and remember, do you need to maintain the homeostasis? Yes. So to maintain the homeostasis, best regulation, right? Best regulation to maintain homeostasis is what? Is negative feedback. क्या बात है? Very good. Very good. Only 3 means 48 clear. Very good, my boss. It's very great performance. I'm just telling you, don't just do anything. Just watch them. These 20 days devote only for the expected PDFs, and you will revise also. You will get the questions also, and obviously you will pass the exam. No need to put effort on any other source, because I have so many students who pass only from the last year session, which was just 10 hours. I think 10 hours I took 100 some question 20, and 10 hours they cleared. So this time so I'm doing almost 100 hours, and the questions are increased to almost 400 500. So daily I discuss around 40-50 questions. So that I'm telling you. Okay, so best regulation to maintain homeostasis is negative feedback. Now what is negative feedback? Negative feedback is basically it bring value back to normal. So what bring the value back to the normal? Answer is negative feedback. Example, let's say our glucose level increase. So our insulin hormone will decrease the glucose till it goes back to normal. So it becomes normal. If there is a decrease in the calcium, which hormone comes? PTH comes, and PTH will increase the calcium to normal value. Okay. Now, if there is an increase in the blood pressure, baroreceptors come, and they will decrease BP to normal. So negative means opposite. So what does negative means? Negative means opposite direction. So remember here, negative means opposite direction. Understood? Opposite direction. So what do? You reverse the value of any given value back to the normal. Like, understood? If there's a high glucose, do you want to make it higher? No, you want to make it less. This is negative. So negative doesn't mean decrease. Negative means opposite. So even an increase in the calcium will be called as negative. What does negative means? It doesn't means decrease. It means opposite. So negative बोले तो opposite direction. Negative बोले तो opposite direction. Are you clear? Have you all understood negative feedback? So negative feedback is what direction? Opposite direction movement. So what's negative feedback? Going in opposite direction. What is opposite? Yes, going from high to low or low to high, so that value become normal. So value become normal. So I hope is clear. Sure. So I'll be ending today's session. Only one page is left. Then we'll be starting with GIT. So next session I will end the remaining one, because I have a class and I have to go and prepare for that. So I'll be ending. We have a 2-hour session today, and I'll be taking this last part of cell physio, like cell junction, in the next session, and then I will be starting with GIT also. So you can watch the lecture in the app till then. Don't waste time कि जब होगा तभी करेंगे. I will take physio, obviously full physio live, but why waste time? When you have time, focus on that, because right now exam time is here, and I have online batch also, offline batch also, expected batch in the evening also. So I'm taking 3 batches simultaneously. So that's why I have less time for our regular new batch. So obviously they will have the classes by other teachers, so they will be giving more because they have more time. I am like taking 3 batches. So I'll be taking from, I think, 12th of July. I'll take longer session, 7-8 hours daily. Till then I'll be focused more on the short sessions. Okay, so chalo, so I'll end the session. Best of luck to all of you. I'll share this PDF, okay, and all of you can use it for your use. Let me save it. So let's meet soon. Take care. Thank you. Bye bye. I'll be sharing the file and ending the session. Chalo, bye bye.